So guys, in the last topic, you have seen how we can make UDP galactose from galactose 1-phosphate. Now let's see what we can do with the UDP galactose, what are its significances as far as the metabolism of galactose is concerned. So one very important function UDP galactose provide the carbon skeleton for many important compounds which we will see. So if we again see the diagram and we focus a little bit on this, which give rise to the formation, in which we show how the UDP galactose is formed. So if you remember it, let's have a little bit of revision of that galactose 1-phosphate is exchanged with UDP glucose, which give rise to the formation of UDP galactose. Now this UDP galactose is an important compound which can give rise to many other compounds also. Keep in mind that the enzyme which is responsible for this exchange is the GALT, G-A-L-T, or galactose 1-phosphate uridylyl transferase. And then once you have the UDP galactose, importantly, this UDP galactose can also be converted back into a reversible reaction into UDP glucose through the enzyme which is what we say the epimerase enzyme. And it's a very significant thing. For instance, under certain circumstances, you can face a condition where you have lack of glucose, and then UDP galactose under the action of epimerase can give rise to UDP glucose, which can be converted into glucose. In this way, UDP galactose is providing a platform where you can get involved into the glycolytic pathway also. That's also a significance of UDP galactose. And not only, glycolytic, other biosynthetic reactions also, which we will see. So UDP galactose, it can serve as the donor of galactose units because it is made up of galactose. These galactose units then can be separated, and then they can be involved in the formation of very important compounds like lactose, like glycoproteins, like glycolipids, like aminoglycans. So these are very important conjugated carbohydrate compounds which are involved in many important functions. These can be structural functions. This can be providing structural framework to the cells. They can be providing the signaling framework to the cell so that cell can perform signaling, and some sort of, we can say, all sorts of activities it is involved. These products can be used. Also, finally one thing I want to share with you, that not only that you can sometimes face circumstances in which glucose is very less, you can also face circumstances in which you don't have much galactose. Let's say you are not being fed with a diet which contains the galactose, and then from where you can get the galactose based metabolism, or where you can get these important compounds which are found from the UDP galactose. Then again, if we move back to the previous slide, and then you can see here that epimerase enzyme is not only giving rise to UDP glucose, but UDP glucose is also converted into UDP galactose epimerase enzyme. And as a result of this, when you are having this problem of galactose unavailability, still through the action of UDP, glucose being converted into UDP galactose, you can get galactose, which can then be used for whatever galactose is doing. For instance, as I have already said you, that you can use it for the formation of certain compounds like lactose, like glycoproteins, and so on. So in this way, the galactose metabolism has its many diverse functions.